Okay, welcome to Simply Psych EDU. This is the second part of the ADHD in Adults series. Today we're talking about the neuroscience of ADHD and answering the question or attempting to answer the question, what do we know about the neurobiology of ADHD? This is a very general overview. This is not a detailed neuroanatomy or neurobiology lecture, but I think this will give you a basic understanding of the neurobio of attention and what we think may be going wrong in individuals who suffer from attention deficit hyperactivity disorder. Unfortunately, I have nothing to disclose financially, so I do not get any money for doing these talks. Medical legal disclosures, the information provided in this presentation is for educational purposes only. The information provided in this talk is for educational purposes and should not be perceived as professional medical advice from the presenter. The information provided in this presentation should not replace the medical advice of your physician. I am not a child-adolescent trained psychiatrist. The opinions of the presenter, me, are my own and do not reflect the opinions of any institution. And the case, which we won't discuss today, but we introduced in the first lecture and we will talk about toward the end, LB, this particular case, the patient was very excited that we were going to discuss her case because she really benefited from treatment and wanted to share her positive response. Some of the diagrams from this talk come from various textbooks. 3 of them are listed here. That's me. Learning objectives. These are for you to review. I'm not going to go over all of them, but I just put them here, and I'll keep putting them there at the beginning of every lecture. Okay, so we're going to start with the neurobiology of attention. Okay, so what is attention? So attention is a cognitive function, and cognition can be conceptualized in a number of different ways, but I think the way that makes the most sense and sort of is the most accepted is that cognition is an individual's ability to, 1, attend to external or internal stimuli, 2, identify the significance of the stimuli, and then 3, to respond appropriately. So attention really describes the mechanism that weighs the importance of these various stimuli and then selects the one that will receive the brain's focus. And we think of it as sort of a component of our level of consciousness, and I think that this may be a little outdated, but when we think of consciousness, there's many different ways to think of consciousness. There are evolving models to think of consciousness, but I think the one that I think makes the most sense and may not be the most accurate, but actually just helps me understand this stuff, is that consciousness really can be broken down into 2 things or 2 parts. So 1, our level of consciousness, basically our how alert, attentive, and aware we are, and then the content of our consciousness, which is really the substrates that together make up our consciousness, like emotions, language, memory, motor planning, executive functioning. So we think of as attention as more of a part of our level of consciousness, whatever that means, but the 2 major functions of attention, now there are many functions, okay, but the 2 major ones that I'm talking about today are selective and directed attention, which involves focusing attention on a particular domain above others, which can be demonstrated by the Stroop Task, which I'll show you in a second. And that selective directed attention is really, it involves many regions of the brain, but primarily what I wanted to point out is that the selective directed attention involves primarily the dorsal anterior cingulate cortex, and that's something that I'll show you in a second. Now, it doesn't mean that that's the only thing, that's involved. Remember that the brain is highly interconnected. When we think of these different regions, we don't think of them as isolated regions that do one or other things. It's more how are these regions functionally connected to one another in circuits, and how how do they interact in a way that allows whatever it is that whatever function that we need, whether it be directed attention or sustained attention. It's just basically these connections between these different parts that ultimately determine our ability to do things at a sort of cognitive level. So the selective directed attention is best illustrated by the Stroop task. There are many other ways you can demonstrate selective or directed attention. Stroop task is just one of them. And then the second major function is the sustained attention, and that's really your ability to sort of maintain concentration on a particular task, and that's best illustrated by the end-back test. There are many different ways, again, to illustrate sustained attention, but a sustained attention primarily involves the dorsolateral prefrontal cortex, but also and its connections essentially to the various regions of the brain, particularly the parietal lobe. Remember that the parietal and the frontal lobe are highly interconnected, as well as subcortical structures, for example, the basal ganglia and the striatum, and then the brainstem. So there are many different connections that are going on here, but I'm just pointing these major regions out, not because I'm saying that they're the only region that's involved, but when we think of dysfunction or we think of lesions in neurologic patients in these various areas, we primarily see dysfunction in either the selective or directed attention or sustained attention. But again, that's a very oversimplified way of looking at the brain, but it's a way to sort of understand it. So this slide is just showing you the neuroanatomy, basic neuroanatomy of what's going on here. And you can see that there's the prefrontal cortex here, right? And the prefrontal cortex it can be divided into different regions. And the dorsolateral prefrontal cortex actually is on the other side. So you can imagine going to the other side over there of the brain, because this brain is cut in half. If you go to the other side, or the right side, if you will, if you see what I'm saying here, that on the other side is the dorsolateral prefrontal cortex. That is primarily involved in sustained attention, and then the dorsal anterior cingulate, this is the anterior cingulate right here. The dorsal part of the anterior cingulate is involved in directed attention. And then, of course, I put in the striatum, or the basal ganglia, which is the caudate and the putamen, this area right here, is also highly connected to those regions as well. So I just wanted to show you this diagram so you had an idea of what brain regions are primarily involved in attention. And then the last thing I'll say is that you can't forget that the parietal lobe is huge when it comes to attentive tasks. And although I don't point it out here, the parietal lobe really is on the lateral side, and it's sort of in this area here, but it's on the lateral side, so it's on the other side of the brain. And so remember that the frontal and the parietal lobes are very important in attentive tasks. So the Stroop tasks is a way of showing or demonstrating selective directed attention. And again, it's not a highly specific test, but I would say that it does a fairly good job of testing someone's selective or directed attention. And basically what the Stroop task is the subjects are shown words of various colors, but not necessarily in the color the word describes. So for example, the word pink would be shown in green, and subjects would have to correctly state that the color of the word was green without stating the actual word pink. And so it takes a little longer to do this than just sort of stating the squares of the colors or just stating what the words are. And so I'm going to show you sort of how this works. So basically, every time you see a word flash on the screen, I just want you to tell me what the color is. So this one's red. Okay, so let's go to the next one. Green. Okay, so that's pretty quick, right? You kind of get it really quick. Blue. Okay, again, it's pretty simple, right? So for a second, when you see this, you see yellow, and the first thing you do is you want to say yellow, but then you stop yourself and you're like, oh, wait a minute, that's purple. And so this sort of the discrepancy between the actual color of the word and the word itself requires more more neural resources, if you will, to be able to identify correctly what the color is. And so in patients who have problems with attention, they may impulsively state the wrong color, or they may take longer to sort of process what color is that. I mean, the delay time may be actually longer. So sustained attention includes functions such as vigilance and concentration, non-distractibility, and in the end-back test, the subject is presented with a sequence of stimuli, and the task consists of indicating when the current stimulus matches the one from n steps earlier in the sequence. So n just means the number of steps back the subject has to remember, and can be adjusted to make the task more or less difficult. So the concept is something similar to a game of memory. And so what you can see here is if you're given n of 2 tests, and the example is this sequence starting with A, and I'm going through this sequence, I'm asking you to identify when that letter is shown on the screen. You have to identify when does that stimulus that you're currently seeing match one that was 2 back before it. So for example, the B here would be one that you'd have to identify because 2 back was also B. Then A you'd have to identify because 2 back was A, and so forth. And so that requires mental effort, obviously, and is a fairly good way to test sustained attention, although there are many other ways to test sustained attention, and I'm not even sure what they're doing currently. This may be very outdated. But this is one way that you can test for sustained attention. In patients who have neurocognitive disorders, particularly patients with schizophrenia or attention deficit disorder or dementia, they have a very hard time with this particular task. Okay, so back to what is attention. So it's important to know that the capacity to concentrate and maintain one's attention correlates with the ability to ignore extraneous stimuli. In other words, your ability to concentrate or to attend to something really requires many things, but 2 of the things that it requires: one, your ability to sort of direct your attention to whatever stimulus is going on that you need to attend to, but also your ability to filter out the extraneous stimuli that are sort of competing for your attention. And so these 2 things sort of need to be happening in order for you to attend to something. And the continuous performance tests, which give an objective estimate of an individual's attention impulsivity, are given to people to sort of test their ability to do these things. And so what we call these CPTs, what they are is basically subjects watch a computer screen and hit a button or click a mouse whenever a specified sequence of symbols or letters appears. And the CPTs reflect the subject's capacity to attend, as well as the ability to restrain impulsive answers. And the results are compared with normative data for individuals in one's age group. And these tests, there's many of them, and these particular neuropsych tests are often administered to patients who are either who have or are suspected of having attention deficit disorder or other neurocognitive disorders that affect your ability to attend to stimuli. And so what's important to know about the CPTs is that when we send somebody for a CPT, we're not sending them, you shouldn't send them as a way to diagnose ADHD. It's not sensitive, these tests aren't sensitive or specific enough to justify its use as a diagnostic tool. In other words, when I'm sending somebody, if I do end up sending somebody for a test, it's to confirm what I already know clinically based on my diagnostic evaluation, because at the end of the day, ADHD is not a diagnosis based on neuropsych testing, it's a diagnosis based on clinical presentation. And the CPT, what it can do is confirm what you sort of already know. Now, just because the test is negative doesn't automatically mean that they don't have it. And so that's something very important. Oftentimes they'll be like, well, I'm not going to even assess you, I'm just going to send you for testing. Well, you may miss 30% of patients. And so that's why I think it's important to know that. All right, so you can see here on the left in this figure A, by the way, these diagrams are taken from The Neuroscience of Clinical Psychiatry, just a wonderful book, which is very, very good for psychiatrists, I think. Really gives you the basics of what you need to know. It's not like a PERVS neuroscience, which goes into much more detail, or Hal Blumenfeld's neuroanatomy through clinical cases. But this particular book, I think, is very good. Anyway, you can see this lady here. She's taking the one of the CPT tests, and then on the right in this graph, what you're seeing is the percent errors on standard CPT tests throughout the lifespan. So you can see that in early age, there are relatively high percent of errors, which means that a young child's ability to attend and refrain from impulsively giving an answer is going to be less, right? They're not as well developed. Their frontal lobe is not as well developed. In fact, their entire cortex is not as well developed. And then as time goes on throughout the lifespan, as you get older, you're going to have less and less errors because your frontal lobe is maturing. And then eventually, once you get to about 60 years old, then your ability to attend on these tasks decrease. So I think this is just a cool diagram showing you throughout the lifespan kind of what happens. And this very much correlates with what's happening neuroanatomically and neurodevelopmentally in terms of how the frontal lobes are maturing. So anyway, I thought that was a cool slide. Okay, so working memory. So working memory describes what is actively being considered at any given moment. Working memory is temporary and it's limited in capacity. And then, working memory and attention are closely related and interdependent. The relationship between working memory and attention is incompletely understood, but may share similar neurobiological mechanisms, which are kind of slowly over time being elucidated. But I think, we're still trying to understand this relationship. If they're the same thing, are they different? Certainly, there's a lot of overlap. And working memory and attention are important components of executive functioning, and executive functioning or executive function includes working memory, attention, and other higher level cognitive skills such as organizing, priorities, and planning. So trauma to the prefrontal cortex impairs working memory. That makes sense, right? If we're saying that parts of the prefrontal cortex are very important for working memory, and they're also very important in the tasks that we just in the tasks like the sustained attention and the Stroop Tasks that I showed you earlier, then it makes sense that if you have trauma to the prefrontal cortex, you're going to have impairment in your working memory. But this was really illustrated, or best illustrated, by the famous case of Phineas Gage, who was a railroad worker who had a tampering iron explode through his frontal cortex. He didn't die, but he went from being this very responsible and organized person to being very impulsive and attentive. And so this is just a picture of him that I got from the internet. But it's a very important case because this was the beginning of really understanding what the frontal lobe and the prefrontal cortex What does it do? What's important about it? I mean, we knew, from animal studies, but in humans, you can't just go around taking people's, traumatizing people's frontal cortex and seeing what happens. I mean, that's unethical. But this particular guy, Phineas Gage, really helped, neuroscientists and clinicians later on understand what are some of the important aspects of the prefrontal cortex. So in the 1970s, neuroscientists began measuring working memory in monkeys. So they implanted microelectrodes. They actually would implant these microelectrodes into individual neurons in the prefrontal cortex. And then they measured activity in these neurons while monkeys were performing what we call a delayed response task. Okay, so this is an example of the delayed response task with the monkey as our subject. So, this monkey is shown these two wells in front of it, and a fruit is placed in one of them, and so the screen obviously is raised, and the monkey observes a piece of fruit that's placed in one of the wells. And then the screen is lowered and the wells are covered. And then after a specific period of time, the screen is raised and the monkey has one chance to remember the correct location of the fruit. So you can see that this is basically a working memory task. So what they found when they did these studies, when they did these delayed response task studies, was that individual neurons responded differently during the delayed response task. So some neurons were active only during the cue response part of the task, while other neurons responded only during the delay period. So, the delayed neurons start firing with the presentation of the cue and stop with the response. These delayed neurons seem to hold the memory of the task. So literally the neural equivalent of working memory, although it's more likely a network of neurons. But when the monkeys incorrectly responded, the delay neurons were usually inactive. So if the monkeys were distracted during the delay period, the delay neurons were less active and the monkeys were found to make incorrect responses or did not respond at all. And so after the studies, the researchers sacrificed the monkeys and identified the location of the microelectrodes. And it turned out that the cue neurons and delay neurons, which I'll show you in a moment, were in different regions and sort of clustered in different areas. And I'll show you here, so that's just a picture of the microelectrodes in the prefrontal cortex of the monkey while they're doing the task. And you'll see here that there's, they were testing the neuronal activity in these different types of neurons. And then when they sacrificed the monkey, they looked at where these microelectrodes were, and they were able to identify these neurons and where they are. And you can see here that the delay neurons are more common than the cue neurons, showing the importance of holding a thought in working memory. Also, it appears that the delay neurons cluster together in what we imagine as part of the network of working memory. And that's what you can see here. So there's clusters. So we can't talk about working memory without talking about two catecholamines, which are dopamine and norepinephrine. And what you see here is a rat with microelectrodes in its brain, and essentially this is the radial maze that it's often used in neuroscience research. And basically what this apparatus is, there's this mouse or this rat or mouse, and there's different treats at different arms of the maze, and the rat or the mouse is allowed to sort of explore and find out where those treats are. They learn the maze, and then the rat or the mouse is then sequestered in this area here and not allowed to see where all the treats are. And then over a period of time, they raise the walls, and then the rat then goes and tries to find where the treats were. And so basically, rat studies using the radial maze demonstrated an inverse correlation between the extracellular dopamine concentration, DA is dopamine, in the prefrontal cortex and the number of errors during the task. So, as there was sort of a reciprocal inverse correlation between how much dopamine there was in the prefrontal cortex and the number of errors, such that when the amount of dopamine was higher, they made less errors, essentially. So that's just showing you that dopamine probably has a very important role in working memory. And then the length of the delay period was also inversely correlated with the extracellular dopamine concentration in the prefrontal cortex and the number of errors. So as the length of the delay period increased, the extracellular dopamine concentration, as the length increased for the delay period, the extracellular concentration of dopamine was decreasing. And so you can see here that is illustrated by this graph here. And this is important because there's implications for vagal nerve stimulation in ADHD. We know that the vagus nerve stimulation activates the locus ceruleus, which is a nucleus within the brainstem where norepinephrine neurons primarily have their cell bodies located. And when you activate the locus ceruleus in the brainstem, it causes widespread release of norepinephrine throughout the cortex. And so if we can pair that vagus nerve stimulation with some type of behavior, we could potentially promote attention to that behavior and cause the brain to change. And so the vagal nerve stimulation is in clinical trials currently for post-stroke rehabilitation and tinnitus, and maybe a future treatment for ADHD, which is just fascinating. Not to mention that many of the medications we use in patients who have attention deficit hyperactivity disorder involve stimulating or enhancing dopaminergic and noradrenergic tone in particular regions of the brain. So when it comes to reward and impulse control, it's important that we discuss this briefly because it has implications for attention deficit hyperactivity disorder, which impulsivity is an important component of the disorder. And controlling the impulse to take an immediate, smaller reward rather than waiting for the larger, delayed reward is essential for completing any project, right? I mean, you're sitting there and you're trying to figure out, well, do I finish what I'm doing now, or do I go to the party that's right down the street? Well, it requires, the impulsive person to be like, screw it, I'm out of here. I'm going to that party. I'm not going to think about it. I'm just going to go. And the sort of more controlled person would say, well, I need to finish this first, and then once I'm done with this, then I can go to the party and enjoy myself. And people who cannot control these impulses or who take the immediate reward first are perpetually falling behind. So an interesting study was done in the 1970s by a Stanford psychologist that I think was really was very interesting and sort of illustrates how reward and impulse control in kids can potentially later on have negative influence or negative consequences. So basically, in the Stanford Marshmallow experiment, they took 4-year-old kids and they sequestered them in a room. And they gave each of the kids one marshmallow. And they were told that, okay, well, you can either eat the marshmallow now, or you can wait until the research assistant who was in the room left to run an errand and came back, and then you could receive 2 marshmallows if you waited. So, we'll give you this one now, and when the research assistant leaves and comes back, which you didn't know how long that would be, if you waited, then you would get 2 marshmallows. And what was interesting is that the children had varying degrees of restraint. And so some children were like, screw it, I'm eating this now because I don't know when this person's coming back, and I'm having this marshmallow right now. And then there was, that was one end of the spectrum. And on the very other end of the spectrum were the kids who just waited for the second marshmallow. So what they did is they determined who were the sort of impulsive eaters and the patient waiters, and then they followed these kids over time into adolescence and adulthood, and they found that the children who were better at inhibiting the impulse to immediately eat that one marshmallow were in general more resilient, confident, and dependable as adolescents, and were more successful students and scored higher on the SAT score. And you can see the average SAT scores there between the impulsive eaters and the patient waiters. Now, you could argue that SAT doesn't really measure anything. Well, if it probably doesn't, but what it does do a good job probably at measuring is sustained attention, a person's ability to sort of sit there and for long periods of time and do mundane, boring tasks. And so I thought that was just an interesting study, and the study is right here at the very bottom if you want to look at that yourself. So the last thing I want to talk about when it comes to reward and impulse control are some of the brain regions, not all of them, right? Not all of the brain regions, but just some of the brain regions that are involved in reward and impulse control. First one, the Nucleus Accumbens. The Nucleus Accumbens is a structure which is located in the ventral striatum, the striatum being the caudate and putamen, which is sort of this earpiece, Bluetooth set looking structure there on both sides of the brain. We know that attention and impulsivity are partially controlled by dopamine within the nucleus accumbens. We know that people are less distracted when pursuing activities they enjoy. Stimulant medications increase dopamine at the nucleus accumbens and improve impulse control. And rats, interestingly, when their nucleus accumbens are damaged, become more impulsive, and they immediately choose the immediate reward instead of waiting. So the nucleus accumbens is very important. Unfortunately, also the nucleus accumbens is important in addictive behaviors and addiction. And most of the drugs of abuse, affect dopamine neurotransmission within the nucleus accumbens. And so this is partly why individuals who use various drugs of abuse, they develop sort of the positive reinforcing effects because of the dopamine that's released within the nucleus accumbens. And that's a partial explanation. It's much more complicated than that, but that's sort of the simplified version. The striatal dopamine transporter density, what the hell is that? Well, the dopamine transporter sits at the very end of the neuron, at the terminal region, and what the dopamine transporter does is it sucks up dopamine after it's released by the neuron. So in other words, when dopamine's released into the synapse, the dopamine's got to go somewhere after it's sort of bound to its receptor and done its job. It's got to go somewhere, right? And so it's either degraded by enzymes. It's degraded by enzymes, or it's diffuses away from the synapse, or it's sucked back into the neuron by the dopamine transporter. And high dopamine transporter density is seen in younger individuals and patients with ADHD, and higher striatal dopamine transporter density has been correlated with more impulsive behavior. Exactly why that is, they're still trying to figure that out. But drug-naive patients with ADHD have a slightly higher density in dopamine transporters. So this is just an interesting graph looking at dopamine transporter density in healthy humans without ADHD, and looking at over the lifespan sort of what happens with the dopamine transporter density in the striatum. And you can see that over time, as people get older, their dopamine transporter density decreases. So dysfunctions in the when it comes to ADHD, neurobiology and neuroscience, dysfunctions in the prefrontal cortex and striatum are the most common abnormal brain findings that have been reported for patients who have ADHD. And Judith Rappaport did a number of neuroimaging studies, and I'm going to talk about, sort of summarize for you what these studies showed. So they have conducted several large prospective case control MRI studies of the brains of children with ADHD, and one study produced multiple MRI scans of 150 children with ADHD and 139 age and sex-matched controls. 60% of the participants had at least 2 scans. So the most interesting finding was that children with ADHD had smaller total brain volumes by approximately 5% compared with controls. The difference held true for all 4 cerebral lobes, including the white and the gray matter, as well as the cerebellum. And the trajectory of the total brain volumes did not change as the children aged, nor was it affected by the use of stimulant medication. So in a follow-up study, Rappaport's group examined another 300 subjects, half of whom had ADHD, and then they measured regional gray matter thickness. And the most unique feature of this second study was that they followed up the clinical outcome as well as the structural changes in the brain of the children over time. And they were able to compare the children who grew out of the disorder with those who did not. And the results showed that children with ADHD had global thinning of all the gray matter compared with controls, although it was most prominent in the prefrontal cortex. Both groups showed the usual pruning of total gray matter as they grew through adolescence. However, 2 regions were unique when correlated with clinical outcome. The children who remained impaired at follow-up had thinner gray matter in the medial prefrontal cortex at the beginning of the study, and children who grew out of the disorder showed a normalization of the gray matter thickness in the right parietal cortex. So these results imply that the normalization of the parietal cortex may be a compensatory mechanism involving recruitment of parietal attention networks. And parietal cortex therefore may play a larger role in attention than previously considered. And I think this is important because when we think of attention nowadays, we really focus not just on the prefrontal cortex, we really focus on the parietal cortex, and the parietal lobe, especially when it comes to disorders where patients have, right hemispheric strokes involving the parietal lobe. They often have what we call hemi-neglect syndrome, which is an attentive problem with the left visual field. Their sensory processing is intact, but their ability to attend to the left visual field is impaired. And so that's just something worth mentioning. So going along with the parietal cortex playing a larger role in attention than we thought, this study compares healthy subjects and patients who have ADHD or suffer from ADHD, and looking at regions of the brain that have more activation during a target detection task. And what you can see is that there are parts of the frontal and parietal lobes that are much more activated in healthy subjects than in ADHD patients during this, a particular attention task. So this is the end of the neurobiology of ADHD. There's much more in the references. You'll find much more reading material if you're interested. This was a very general overview, and by no means was it all inclusive. I don't have time to go into all of the neuroanatomy and neuro studies, but I think that if you are interested, I will make sure that in the references at the very, very end, you can look through those references and read the studies if you're interested.